Erstmal meinen Bildschirm teilen. Ja. Okay. Ja, also danke, dass ich hier vortragen kann. Mache ich für euch doch immer gerne, ist doch klar. So, and yeah, Peter asked me to present our latest research data from Berlin. So it's the data that comes from a collaboration from the Charité and the Max Planck Institute. And I think it's a nice example of how clinical work and lab work can be merged to achieve something that neither group could have achieved without the other. So this is, yeah, this was lucky for all participants. The title is Aberrant phase separation and nucleolar dysfunction in rare genetic diseases. So how does the story start? Back in 2021, here at our institute, we identified an interesting variant in a gene called HMGB1. We've seen this variant in a fetus with a complex limb malformation, and it's been a de novo deletion of 4 base pairs that resulted in a frame shift. And this frame shift occurred in the final axon of HMGB1. Therefore, we assume that it would not undergo nonsense-mediated decay, but would escape NMD. And this frame shift occurred here. So what do you see here? This is human is the reference amino acid sequence at the C-terminus of HMGB1. And what you can see is that it's largely comprised of aspartic and glutamic acid. So this is an E and D repeat. Aspartic and glutamic acids are the two, as you all know, the 2 amino acids that have very acidic residues. And the frame shift occurred here at the very, so close to the beginning of this E and D repeat. And this turned this sequence into that sequence that you can see here. And I think what's obvious is that it immediately caught our attention, because now the acidic character of the C-terminus was lost. We checked whether the C-terminus is evolutionarily conserved, and indeed it is. So here you see the sequences of mouse, cat, chicken, and so on. And down to the zebrafish, this is an E and D repeat. And it's also well characterized in the literature. HMGB1 is a protein that has various functions. It belongs to a very old, evolutionarily old group of proteins, the homeobox domain containing group member family. And this E and D repeat is known to mediate the interaction of HMGB1 and DNA. So at this time in 2021, we knew we had this de novo mutation. We knew it was conserved. We knew our patient was grossly malformed. It was a fetus. He didn't even survive in pregnancy. And we were pretty sure that this is the cause. However, we lacked a second family. But I think it's obvious why we still thought, okay, it must be the cause. And when we published this paper, my boss, Professor Horn, she reviewed this paper for us, and she gave us a comment that what we called a complex limb malformation, she saw a syndrome in that she could recognize. We couldn't. So this is the patient, the fetus. And as you can see, the lower limbs are grossly malformed. That's because the tibia is missing. What's not so obvious on this photograph is there are too many toes, by far too many, not only 6, but 8. Then there's pterygia, so the fetus could not extend the upper arms properly. Also, the fetus was too small. There's a genitourinary malformation. Here you can see the pterygia. So this was grossly malformed, and we thought, okay, this is a complex limb malformation. Here there are the radiograms, and yeah, there you can see the additional toes on both sides. So it closely resembled mirror image polydactyly. We thought it's a type of mirror image polydactyly. And what you can see is that there's only one long bone in the lower limb. That's the femur. And in the lower legs, there is only one bone. We were unsure whether this is the tibia or the fibula. Either way would be too short, and the other bone was missing. And at this time, we've seen another patient. So I didn't see him, but he was seen in Schleswig-Holstein by the colleagues there. And they also contacted Denise Horn asking for her advice. And that's how she realized these two patients, they might have the same condition. You see that there are the additional toes. Also here, this patient is from, genetically, it's a male. However, the genitalia are intersexual. And we saw these malformations at the fingers. It's as if the nails are coming from the center of the finger. They are not positioned on either side. And that's because the hands, they appear as if both sides of these hands are palmar sides. So he has no dorsal side on the hands. And Denise knew, or she suggested, that this is a recognized syndrome described in 1997, which is called BPTA syndrome, brachyphalangy, polydactyly, and tibial aplasia syndrome. And here you can see the first patient who was ever presented to have this condition. And indeed, there are similar findings. So the number of toes is increased. You can see that the lower limbs are malformed, similarly to our patients. And we then searched the literature, and there are only very few such cases. So without our newly identified patients, there have been only 9 patients in the literature featuring BPTA syndrome. And when you have a look at them, you will realize, I think we all agree that they definitely have the very same condition. So it's a striking phenotype. However, you need to know it. And that's something we didn't recognize, because I personally and the others who were involved, apart from Denise Horn, we didn't knew that condition because it's so ultra rare, but she knew. And once we realized that this is a condition, and we have one patient here in Berlin and another in Schleswig-Holstein, we started to see whether we could more DNA samples of these patients. And indeed, this patient, who was presented in 2010 in the literature, we had a blood sample of him, because Professor Mundlos, he collects blood samples of patients with lymph malformations in the Berlin Lymph Study, and we were lucky, we had a sample of this very patient. This slide, to be sure that they all had the same condition, you know that I learned from Peter, and that's why I'm always using face-to-gene to prove that a syndrome has a characteristic face, and they all share the same facial characteristics. And this can be caught by deep gestalt, not because it will not suggest this disease, BPTA syndrome, because it's not trained for it, but it suggests something else, blepharophimosis, ptosis, and epicanthus inversus syndrome. And this is something that deep gestalt would only very rarely suggest. And in these patients, in nearly all of them, it was suggested. That's why we also show that they share a facial characteristic, which further reassured us that they have the same condition. So we checked our database, and we realized that there was another patient seen in Germany, here presented as individual 2, who also featured BPTA syndrome. And during the time this paper was written, another patient was born, and we were contacted, which became individual 4. And now that we had a DNA of 5 individuals, we just sequenced them. So family 5 was already identified. That's the family you've already seen. And now we have 4 additional families, and we knew what we were looking for. We wanted to see whether they all had this mutation in HMGB1. And what you can see here is, indeed, these 4 families, they had the very same de novo variant. And this family had a very similar de novo variant, also a frameshift at the beginning of the acidic tail, only 2 codons more upstream. Individual 1 that was seen in Schleswig-Holstein was luckily also genome sequenced. That's why we could rule out that there was any other mutation potentially causing this condition. And so this just illustrates how our filtration steps. So we started with all the variants called, and then I think we do it as everyone does it. We check for, first we check for coding variants, then for those coding variants that are loss of function variants, and those that are rare, those that are truncating, affecting genes with a PLI greater 0.8, and then we filter for de novo variants. And in both individuals, in our individual here in Berlin, as well as in the individual one from Schleswig-Holstein, there was only a single variant that met all these criteria, and that was the HMGB1 frameshift. Individual 4 received exome sequencing, and there again, we saw only a single variant that met all these criteria, and that was HMGB1. So now you've already seen the reference amino acid sequence of HMGB1. Here we now not only shaded acidic residues in red, but also shaded basic residues in blue. And in NOMAD, there are 280,000 reference alleles for HMGB1, but only 1,123 non-synonymous variants in HMGB1's acidic tail. So variants that are positioned somewhere here. And of these non-synonymous variants, the majority used to be deletions, and a few were acidic substitutions, and even less were insertions of acidic residues, and only very few were non-acidic substitutions. So let's start with the insertions. So 5 alleles distributed among 3 variants are insertions, and these insertions only insert acidic amino acids. So they wouldn't change the acidic nature of this C-terminal tail. Here you see the deletions and substitutions. Let's first have a look at the deletions. So when there is a dash here, this means that the respective amino acid is just missing from the tail. And what you see is that these 906 variants are distributed among these alleles highlighted here in black. And they would shorten the C-terminus, but they again would not change the acidic nature of this C-terminal tail. Some are substitutions, those 207, but they only turn glutamic acid into aspartic acid or the other way around. So they again do not change the acidic nature of that tail. Then there are 5 variants in NOMAD that are non-acidic and no deletions. They are distributed among 2 alleles, 2 variants, those 2. One is here and the other is there. They are not basic. We are not, so I would think they do not harm the acidic nature of this tail. Anyway, they are super rare. This reaffirmed us that this change of the acidic nature of the C-terminus of HMGB1 is pathogenic. HMGB1 was just recently, during the time we were writing our paper, HMGB1 was identified to be associated with another condition, a certain neurodevelopmental phenotype. It's rather unspecific, and the main characteristics are neurodevelopmental delay. What is important is these other variants do not cause skeletal malformations in the individuals. And the paper that described them showed point or small variants within the HMGB1, but none of them was within the C-terminus in the acidic tail. So here shaded in gray are the HMG boxes of HMGB1. So this is the HMG box 1, and that's the HMG box 2, sometimes also referred to as HMG box A and HMG box B. And what you see here is that there are frameshifts among them, but these frameshifts are positioned not in the ultimate, but in earlier axons. Therefore, we assume that they lead to the degradation of the HMGB1 mRNA due to nonsense-mediated decay. Also, there are substitutions, but they are positioned within these functionally relevant domains of HMGB1. Our mutations, however, are positioned here at the beginning of the C-terminus, and they, I've already mentioned it, they change the acidic nature of this C-terminus to a basic tail. Now, when we shade these amino acids in blue when they are basic, then you see that it's not only that the acidic nature is lost, but what replaces the C-terminus is now basic. And also, there is this, this leads to an altered position of the stop codon, the frameshift. So the stop codon now occurs here, and that's why this tail is a bit longer. And there is a hydrophobic patch to this tail. So these amino acids, they have a hydrophobic character. So until here, to me, that was just clinical genetics, how we all do it. And then Stefan Mundlos said he had heard a talk about protein domains that do not have a certain shape. I've never heard of this before I started in this project. Usually I would have thought that protein domains are either beta sheets or alpha helices or beta turns, something like this, but they usually assume a certain shape. However, this is not true. There is something that's called an intrinsically disordered region. That means the amino acids, the sequence of these amino acids, does not have a propensity to assume a certain shape. It remains disordered. In other words, they do not have they do not assume a secondary structure, a defined secondary structure. They remain unstructured. And HMGB1's acidic tail is such an intrinsically disordered region. Stefan Mundlos guessed this because there's this repeat of only two kinds of amino acids. And IDRs, intrinsically disordered regions, are often caused when one or just two amino acids are repeated. This is likely to cause an IDR. And he contacted our colleagues at the MPI, the group of Danish Nies, and they are experts on intrinsically disordered regions. And now that we were sure that these mutations caused brachyphalangy, polydactyly, tibial aplasia syndrome, we were intrigued to know how it does so. And we wanted to know what is happening here functionally. And the colleagues there, you can bioinformatically predict the propensity of an amino acid sequence to be disordered or ordered. And what you see here is that their prediction told them that the disordered character of the wild type amino acid sequence, here showed it as a dashed black line, is now changed to an ordered or a more ordered structure at the very end here, but remains disordered in the beginning. And this resembles what we see here. So this is disordered, then this part is also disordered in those newly formed C-terminal tails, but this hydrophobic patch, that appears to be ordered. One thing that I missed is those two frameshifts that we identified. They result in it, but maybe you've all seen it from the image in a way, they result in the very same amino acid sequence. They only differ on the amino acid level by a single amino acid. This can also be seen, those disordered regions can also be seen in predictions from the AlphaFold neural network. So this is a prediction of HMGB1's spatial structure made with AlphaFold. And what you see is, what we all know, these typically structures, alpha helices, for example, here, so ordered regions. But what you also see is disordered regions. Here, that would be a disordered region because it is not displayed as an alpha helix. That's how AlphaFold tells you it's disordered. And also have a look at the color. AlphaFold's confidence score is depicted in a color scheme. When AlphaFold is sure that a certain spatial structure is assumed, this is depicted in blue. When it's unsure, it's depicted in a more warm color. So the color tells you AlphaFold is not able to predict the structure. This incapacity of AlphaFold to predict a certain structure can be interpreted as the structure to be disordered. AlphaFold cannot predict it because it has no structure. So here we see what we also saw here, that the wild type HMGB1, its C-terminus, is intrinsically disordered. This is the prediction of AlphaFold of our mutant HMGB1. And what you see is now AlphaFold predicts that this intrinsically disordered region is changed. AlphaFold assumes that it has now an alpha helical structure. This is contrasted by this ponder prediction, where the prediction says it still remains intrinsically disordered. We tested this experimentally. We think also the altered, the mutated HMGB1 is intrinsically disordered in its tail, and only the very end of the mutant HMGB1 is ordered. So why does this matter, whether a domain is ordered or disordered? Intrinsically disordered regions help proteins to form condensates. They do this by unspecific interactions, and this unspecific character of these interactions is guaranteed because these domains are disordered. And this formation of condensates is necessary for a process that's called phase separation. You know this process. You know it from when you try to mix oil and water. This will not work. So you can perturb your mixture, and then you put your glass on your desk, you wait for a few moments, and then you have two phases, an oily phase and a water phase. And they, those two phases, they separate on their own because of the biochemical features of the molecules within. And proteins can also do this. I learned in med school that you always need a lipid double layer structure to separate organelles from one another. But this is not the full picture. Proteins can also achieve that very task. And there's one organelle that has no membrane. I knew it that it has no membrane, but I personally never thought about it. I always said, okay, yeah, it has no membrane, so what? But that's very interesting, and that's the nucleolus. The nucleolus is separated from the rest of the nucleus, but there's no nucleolar membrane. Something like this does not exist. So how can the nucleolus be separated from the rest of the nucleus? And that's because molecules, proteins within the nucleolus have phase separating functions, and these are encoded in their intrinsically disordered regions. Now, this said, what did we test? We tested whether this change of, and that's something that not I did, but the colleagues at the MPI, especially my colleague Henry Niskanen did it. What they did is they tested whether the phase separating capacities of mutant HMGB1 differ from wild type HMGB1. First, look here at this series of images, the wild type HMGB1. For those biomolecular condensates to be formed, there needs to be a certain concentration of the protein. And what they did is they just created higher and higher concentrations of HMGB1. And you see that around 5 micromolar, then at this concentration, you see condensates that are formed. Maybe already at 2 micromolar, there's one. And at 10, they're properly seen. Now, in the mutant HMGB1, this happens at already at lower concentrations. Here we start to see condensates. And at those concentrations that would be required in wild type HMGB1, we see by far more condensates. And something else that I learned is when I saw this image for the first time, I was like, yes, okay, I get the idea. So there's something earlier than here. But it's not only that the concentration required to form condensates is diminished, also the shape of the condensates that are formed differs. Here, the condensates are rather spherical in shape. We could refer to them as droplets. Here, they are not this spherical anymore. So these condensates obviously also have another nature. And that's something that I also only learned during that process. That's something that's important to those people who are doing research in phase separation. Is it spherical? Is it not spherical? And so on. These graphs show us, so here you can see this, what you already got from this series of images, that the concentration required for condensate formation is lower in wild type HMGB1. And here, this is also a very nice experiment, what they did. They performed a FRAP experiment, FRAP, F-R-A-P. That means fluorescence recovery after photobleaching. And what you do is you take an emulsion of the protein you want to investigate, and then you bleach a part of it with a high intensity light signal. And the fluorescence in that region that you've bleached is lost. And you wait, you just wait. And what happens is, as the molecules diffuse, the fluorescence is recovered. And when those molecules diffuse quickly, the fluorescent signal recovers quickly. When the diffusion rate is diminished, then the fluorescent signal in the region that you've bleached will not recover that quickly. And this means what we see here is that the diffusion rate of HMGB1, mutant HMGB1, is diminished. So what did the colleagues also do? They wanted to see where does HMGB1 usually occur in a cell, and where is it now as it is mutated. Wild type HMGB1 shows, so this is maybe because that's something that I also needed to understand. If you took an image of this sphere here, then you would end up with this image. So this is one such condensate. And they tagged HMGB1 in green and mediator complex, which is a protein required in transcription, in purple. And you see that the distribution of HMGB1 and wild type HMGB1 and mediator complex in these condensates is the same. So obviously HMGB1 is where mediator complex is, usually. Now, the mutant HMGB1 shows a different pattern. So this is where mediator complex is. This is, now we see in the center, we lose signal. At the borders, there is some signal of HMGB1, mutant HMGB1 still, but it's not equally distributed throughout that condensate as mediator complex would have been. So this confirms that the phase separating nature of HMGB1 is altered in mutant HMGB1. And then they used another control, not mediator complex, but NPM1. And NPM1 is a protein that's present in the nucleolus, in the granular component of the nucleolus. And I will have a slide that explains what that is, but now just think of it as a marker for the nucleolus. And usually, where we see NPM1 signal, we barely see, if not to say nearly nothing, we barely see wild type HMGB1 signal. However, mutant HMGB1 has a stronger signal there. And this means mutant HMGB1 is not where transcription takes place anymore, here, but now it's within the nucleolus. So it changed its position. They also checked whether a mutant HMGB1 appeared in condensates that usually would resemble heterochromatin. This is the case. Usually it's not there. And now in these condensates, it appears, but again, at the border of the condensates. So here, maybe we should start with this. This is in the nucleolus. And the nucleolus, I mentioned it, is an organelle that has no membrane. It is, I thought that's very fascinating, a droplet within a droplet within a droplet. And that's because these proteins, NPM1, FIP1, and R1, they have different phase separating capacities. And that's why they assume on their own this structure, a droplet in a droplet in a droplet. And let me go back. We have seen here that mutant HMGB1 is now where NPM1 usually is. This means now mutant HMGB1 is in this outer layer of the nucleolus. That's where it now appears. And let's go on. And they could show this, and then they wondered, how does it, the one here. So then they wondered, does it also interact with FIP1? And because FIP1 would be the droplet within the NPM1 droplet. And what you see here, maybe that's the image that you should draw your attention to. What you see here is the green signal is outside, the purple signal inside. So this really means HMGB1 is within the nucleolus, but at its outer layer, just like NPM1 would be. It's in the granular component of the nucleolus. So now we were sure that this is the pathomechanism at play. So you change the IDR. Now this changes this phase-separating behavior of the molecule. Therefore, the molecule is positioned in another phase of the cell. And as this happens, disease occurs. But we wondered, is this something rare, something only possible in BPTAS, or is this a mechanism that we should, as geneticists, think of more frequently? And we searched different databases, ClinVar, Cosmic, so databases that contain pathogenic variants. And we searched for variants that are frameshifts that are in protein domains at the C terminus, which are predicted to be intrinsically disordered. And indeed, these frameshifts exist. They have been described. They already have also been described as pathogenic. And but nobody, well, maybe nobody, but only very few people so far thought of the functional implications of these variants. So yeah, here, this should represent our catalog. So we try to put it in a figure that's easier to visualize, to visually assess what we found. So all those genes that you see here contain at least one frameshift that meets this criteria, a frameshift at the C terminus that changes an IDR. Okay. So there were several. And the colleagues at the MPI, what they then did is they took some of these variants and cloned them into their U2OS cells. So they did the same what they've done for our variant, our BP-TAS causing variant, for those other variants, to see whether they see similar patterns here. And indeed, they see this. So here, encircled here, is a nucleus, not a nucleolus, but a nucleus. So this is a nucleus. And now, usually wild type HMGB3 has a similar structure as HMGB1, is dispersed throughout that nucleus. But mutant, when we took this frameshift that we identified through these databases, a mutant HMGB3 forms smaller condensates. And again, they then tested whether it's in the, so FRIP1, so in the granular component, or in the, let me go back, in the dense fibrillar component. And we see similar patterns. I think it's obvious from these images. We see similar patterns as we have seen in HMGB1 in all these other variants. So always these frameshifts change the phase separating capacity of the affected proteins. And this changes their positioning within the cell. And of course, this has a functional implication for their role in disease. I think this the example that I personally considered most striking is this one. DEVIL1 is a gene that's associated with Robinow syndrome. That is known, and it's known for quite a time now. However, when you plot the known disease-causing variants of DEVIL1 on the entire gene, you see that they cluster here, and you also see that most of them, if not to say all of them, are frameshifts. So now we wonder, how is this? Why is there no missense variant in DEVIL1 causing Robinow syndrome? And the answer to us is now, what happens here is we have at the C terminus, we have a an intrinsically disordered region. When a frameshift occurs at the beginning of that disordered region, it changes this disordered region. Therefore, it changes the phase separating capacity of DAVL1, and this is what leads to Robinot syndrome. So this frameshift-induced IDR swapping, that's a new disease mechanism, and that's what made this finding so interesting. So to conclude, frameshift-induced IDR swaps in HMGB1 cause brachyphalangy, polydactyly, tibial aplasia syndrome. They disturb the nucleolar physiology by altered phase separation capacity, and they are probably, this is probably a frequent pathomechanism in congenital syndromes. If you want to learn more about it, then check our manuscript. And I thank everyone who was involved, because there was a huge collaboration. We couldn't have done it alone, especially I think Henry Niskanen, who is the biochemist at the MPI here in Berlin and who did all the experiments. Alex created this, this catalog. Stefan Mundlos already mentioned. Almut Kalibe was the one who, together with Denise Horn, realized that these patients had BPTAS, which just enabled us to identify more patients. And Dennis Nies, he is the head of the lab of Henry at the MPI, and Malte was the one who, together with me, browsed through all these limb malformation patients to identify this family. Thank you.
Yeah, cool. Thanks. If you have a patient with tibia hypoplasia and check OMIM, you find LMBR1. Any connection to that gene?
No. So within LMBR1, there's the ZRS element. And I think what they share, those two phenotypes, is the polydactyly. We know that ZRS also causes mirror image polydactyly, and limb malformation. But I wouldn't see whether, actually, I don't know. I would need to check whether LMBR1 contains an intrinsically disordered tail. I wouldn't think so.
Probably not the same mechanism. I'm just surprised. It seems to be, as you said, a general mechanism of organizing proteins, and it's disturbed. And surprise, surprise, it's a quite specific skeletal malformation. That's why I wondered whether there is any interaction with yet other genes that are known to cause that phenotype, which might be compromised.
Yeah, a similar question was asked by the reviewers, and that is, as we've shown that on the functional, on the cellular and molecular level, all these mutations have a similar effect. Why do they cause different syndromes? And we think that is because, as always in genetics, these genes are expressed at different times in different organs, and those and the proteins have different functions. And that's why we see on a clinical level another syndrome, while the phenotype, if you want to refer to this as a phenotype, the nucleolar phenotype is similar. The overall clinical phenotype is not.
Oh, ja.
Okay, kann ich in Deutsch fragen?
Ja.
Glaubt ihr, dass das eher mit Malformationssyndromen zu tun hat und weniger mit Neurodevelopmental Delay? Also würdet ihr sagen, dass da dass es da schon Unterschiede gibt in der Erkrankungsgruppe, in das eher gehört?
Überhaupt nicht. Also wir glauben gar nicht, dass es Unterschiede gibt in der Erkrankungsgruppe, sondern wir denken wirklich, es hängt davon ab, welches Gen betroffen ist. So ähnlich wie wenn man würde ja auch nicht sagen, ein Frameshift macht immer eine Skelettmalformation. Ein Frameshift kann auch alles machen. Und genauso glauben wir, dass ein IDR-Swap grundsätzlich jede Erkrankung hervorrufen kann. Entscheidend ist, in welchem Gen er auftritt und welche Rolle das Genprodukt dann hat. Wir glauben auch, dass es nicht nur relevant ist für angeborene Fehlbildungssyndrome, sondern auch Implikationen natürlich gibt für Tumore und Krebserkrankungen. Das konnten wir auch daran zeigen, dass dieser Katalog, den wir gebildet haben, da haben wir eben nicht nur ClinVar genutzt, sondern unter anderem auch Cosmic, die Cosmic-Datenbank, und die enthält Mutationen, die in Tumoren beschrieben sind.
Danke.
Axel? Du bist noch stumm.
Ja, vielen Dank. Also ich bin einmal neugierig bei heterozygoten, also die Varianten sind ja heterozygot und die Experimente, das war ja praktisch dann durchgängig.
Ja.
Und wie stellt man sich das vor?
Wir gehen davon aus, dass wenn diese Variante beim Menschen homozygot aufträte, dass das mit dem Leben nicht vereinbar ist. Das wissen wir daher, dass wenn man HMGB1 in Mäusen komplett ausnockt, dann ist es auch mit dem Leben nicht vereinbar. Die Experimente, die gemacht wurden, waren in Zelllinien, also in diesen U2-OS-Zellen, und da wurden die reinkloniert und dadurch sind die dann tatsächlich homozygot. Deswegen kann man die Bilder, die ich hier gezeigt habe, nicht 1:1 übertragen auf das Geschehen, was in den Patienten tatsächlich passiert. Aber was man daraus herauslesen kann, ist, dass diese Phasentrennungseigenschaften sich verändert haben. Und es wäre ja auch, das hätte ich vielleicht noch deutlicher sagen müssen, es wäre ja auch sonst, wenn man sich vorstellt, HMGB1-Mutanten würden schon im heterozygoten Zustand dazu führen, dass der Nucleolus komplett zerstört ist, wäre das ja auch gar nicht mit dem Leben vereinbar. Sondern die Experimente sollten vor allen Dingen prüfen und haben das auch meiner Meinung nach gut getan, ob die Phasentrennungseigenschaften verändert sind. Und das ist sie. Ja.
Ich dachte, du stellst jetzt eine Frage über AlphaFold, Axel. Weil das würde ich gerne wissen, was meinst du, wie es jetzt mit den Prediction Tools weitergeht? Das sieht ja wirklich so aus, als wenn man das eigentlich ganz gut modeln können müsste. Also ein Loss of IDR.
Ja, das denke ich auch. Also wir konnten es zusammen mit den Bioinformatikern vom MPI relativ gut machen. Sicherlich ist unser Katalog noch nicht das Ende der Geschichte. Ja, den kann man auf jeden Fall noch genauer machen, noch bessere Prediction Tools mit reinnehmen. Also das kann nur der Anfang sein. Aber auch ich denke, dass man diese IDRs gut vorhersagen kann. Und es gibt, also ich kenne jetzt 2 Vorhersagesysteme. Das eine ist dieser Ponder-Score, das was dann so als Graph rauskommt. Und das andere ist eben, dass es in AlphaFold selbst enthalten ist. Und das ist ganz witzig, das ist eigentlich wie ein Byproduct von AlphaFold. Ne? AlphaFold versucht ja immer was vorherzusagen und dann sagt es im Grunde, ja, ich weiß auch nicht, welche Form das annimmt. Okay. Und dann hat man festgestellt, naja, weil du das gar nicht lernen konntest, weil es gar keine Form annimmt. Ne? Genau.
Okay. Tim?
Ja, vielleicht, also ich hatte eine andere Frage, aber wenn wir kurz bei AlphaFold bleiben, glaubst du, dass dieses, ich weiß nicht, was das macht, immer dann so eine Region, so eine IDR-Region ist?
Nee,
oder? Okay,
nee, dass es das immer ist, glaube ich nicht, aber ich glaube, es ist sehr häufig.
Okay.
Ja.
Und die andere Frage, die ich mir gestellt habe, am Anfang bei der Übersicht, wo du auf die Neurodevelopmental Delay-Erkrankung eingehst mit den mehr N-terminalen Varianten, da war eine relativ späte DUB-A, wo ich mich gefragt habe, ob die noch NMD macht oder ob das ein gutes Model wäre, um zu gucken, was passiert denn, wenn ich keinen Swap habe, sondern das Ding einfach weg ist.
Ja, gut gesehen. Da ist es, und die haben wir in unserem Paper auch nochmal extra diskutiert, da ist es tatsächlich so, die ist in dem vorletzten Exon, sodass wir nicht sicher sagen können, ob dass die in Nonsense-Mediated Decay geht. Wir glauben, dass sie aus 2 Gründen diesen Neurodevelopmental Phenotype macht und nicht BPTAS. Das eine ist, dass vor allem, weil sie hier diese HMG-Box mit wegnimmt. Und die hat eine Funktion. Und wir sind ziemlich sicher, was man ja zum Beispiel aus dieser Substitution weiß, wenn man dort rumfummelt an der HMG-Box, dann passiert was anderes. Außerdem führt sie zu einem früheren Stopp, sodass das hier weg ist und nicht getauscht ist. Das sind Daten, die habe ich jetzt in diesem Vortrag nicht gezeigt. Das haben die Kollegen am MP alles rauf und runter getestet. Und sie haben auch geschaut, was passiert. Sieht man's? Sie haben auch geschaut, was passiert, wenn man diesen IDR einfach wegnimmt. Und sie haben auch geschaut, was passiert, wenn man hier hinten diesen hydrophoben Teil nicht hinzunimmt. Und tatsächlich passieren dann jeweils andere Dinge. Und auch das macht diese Mutation so interessant und diesen Fall so interessant, weil er grundsätzlich Informationen über diese Phasentrennungsgeschichten uns gibt. Denn bisher ist es so, wenn man sich dazu beliest, also 50% aller Proteine haben mindestens eine IDR, die allermeisten Transkriptionsfaktoren haben eine IDR, aber man hat noch nicht so ganz verstanden, wie diese IDRs miteinander interagieren und warum zum Beispiel Nochmal hier vorgehen. Warum zum Beispiel hier diese 3 Phasen möglich sind. Also ich habe mir das ja immer bisher so erklärt, es gibt 2 Phasen grundsätzlich, Öl und Wasser. Hat man irgendwann mal als Kind gelernt und das ist es. Aber das kann ja gar nicht stimmen, weil hier allein schon 3 Phasen auftauchen. Und jetzt ist die Frage, wie können diese Proteine mehr als diese 2 Phasen hervorrufen? Offensichtlich gibt es Sequenzeigenschaften in den Proteinen, die dann doch nicht so unspezifisch sind und ihnen sagen, okay, du gehörst zu Phase 1, du gehörst zu Phase 2, du gehörst zu Phase 3, vielleicht gibt es auch noch 4, 5, 6. Und da fehlt uns, die Leute, die in der Phase Separation forschen, die nennen das eine Grammatik. Die sagen immer, wir haben keine Grammatik. Ich als Genetiker tendiere immer dazu zu sagen, ja, uns fehlt da der Code. Ja? Also es gibt keine Grammatik dafür. Wir können nicht aus den Aminosäurefolgen zum jetzigen Zeitpunkt vorhersagen, mit wem diese Proteine gerne in eine Phase gehen. Und dieses hier ist es gibt jetzt gerade ein paar weitere Arbeiten, die noch erschienen sind, zeigt uns, wenn man von sauer nach basisch geht und dann noch einen hydrophoben kleinen Rest dran macht, dann ist es die Grammatik oder der Befehl für, geh in die granuläre Komponente. Das konnte man hier raussehen. Das ist doch nicht die ganze Grammatik der Phasentrennung, aber es ist wie, als würden wir ein Codon übersetzen können. Und da gibt es ein paar andere Paper, die haben noch so ein paar andere Sachen gemacht, die machen dann so Sachen wie immer sauer, basisch, sauer, basisch, sauer, basisch und dann gucken sie, was dann passiert. Also da passiert gerade viel und da muss noch viel geforscht werden. Ja. Aber, um auf deine Frage zurückzukommen, die Veränderung, die dieser Frameshift hat, würde in einem anderen Befehl folgen, weil eine andere hier, dieser IDA dann anders aussehe und damit die Grammatik anders anzuwenden wäre. Aber das Wissen dafür reicht halt noch nicht aus, um das vorher zu sagen. Deswegen macht es nicht das Gleiche.
Ja, die IDA ist weg, ne?
Genau, ja, die IDA ist weg. Ja.
Ja. Danke.
Also, dass man da jetzt zur Interpretation Grammatikkenntnisse braucht, das finde ich ja schon ein bisschen abschreckend, weil ich wollte eigentlich fragen, ob du glaubst, dass das auf Kids hinausläuft, die sich dann jedes Labor bestellt, wenn es mal so einen Patienten hat, oder dass man da spezialisierte Gruppen hat, mit denen man dann...
Meinst du jetzt zu Forschungszwecken oder zu therapeutischen Zwecken?
Nö, also jetzt erstmal, um es diagnostisch fix zu machen. Wahrscheinlich will man schon für die nächsten, die jetzt mit
genau,
publiziert werden,
also da denke ich, sind die Assays, so wie sie hier im Niche Lab am MPE etabliert wurden, super geeignet. Also die haben die ja nicht erfunden, diese Assays. Wenn man andere Paper anschaut, sieht man, das ist, wie es zurzeit gemacht wird. Wenn man IDAs untersucht, dann macht man eben man kloniert das rein in die U2S-Zelle, man macht so ein FRAP-Experiment, man guckt, welche Proteine, von denen man schon weiß, dass sie bestimmte Phasentrennungseigenschaften haben, nutzt man als Kontrolle, man färbt die in unterschiedlichen Farben und so weiter. Das wäre immer das gleiche Vorgehen. Ja, also das geeignete Vorgehen. Ja. Wir werden dann häufig auch gefragt, deswegen habe ich gerade gefragt, wegen Forschung oder Therapie, häufig werde ich dann gefragt, ja, jetzt wo man weiß, was das macht und wie es zu der Krankheit führt, muss man doch quasi sofort schon ein Medikament entwickeln können. Und da muss ich immer schmunzeln, da freue ich mich immer, wenn ich dann hier zu den Genetikern spreche. Ja, so einfach ist es nicht, weil wir müssten das ja wissen, bevor der Patient geboren ist und wahrscheinlich noch kurz nachdem der gezeugt worden ist und dann noch irgendwie am besten präimplantationell da Interventionen machen mit irgendeinem Molekül. Das wird nicht gehen. Also dieses Problem löst auch das Wissen um die Phasentrennung nicht. Da denke ich tatsächlich, dass es sich, wenn es sich überhaupt für therapeutische Zwecke eignet, dafür ist es ja noch viel in den Kinderschuhen, um da zu wissen, wie man das jetzt überhaupt beeinflussen kann, aber wenn es sich dafür eignet, dann eher zur Krebstherapie, aber nicht zur Behandlung von erblichen Fehlbildungen.
Ja. Ja, spannend. Vielen Dank für den Vortrag. Den haben wir jetzt, glaube ich, jetzt haben wir es deutlich ausführlicher verstehen können als auf der GFH. Aber da,
ja, da hat man immer nur 10 Minuten.
Ja.
Mal gucken, wie ich das mache. Okay.
Ja, super. Gibt es noch Fragen? Dann